Hello everyone, and thanks for coming to my talk, Prep and Check Proteins, AAVs, and LNPs with Stunner, Uncle, and Big Tuna. When it comes to biomolecules, no matter what you're working on, there's going to be a challenge with sample prep. For proteins and antibodies, exploring the formulation space for the optimal formulation means that you could be testing tens or hundreds or even thousands of combinations of proteins and formulations. For viral vectors, like AAV, you'll often have to concentrate very dilute viral particles to get to your final concentration and in your final formulation. For lipid nanoparticles, or LNPs, you'll have to remove organic solvent, like ethanol, very quickly after production to ensure your LNPs stay stable. Thankfully, there's the Unchained Labs Gene Therapy Squad that's here to help you prep and check all of these biomolecules. Big Tuna is our automated buffer exchange platform that also allows you to do sample cleanup or concentration. Stunner is our perfect benchtop quant tool that uses UV-Vis and DLS to look at both concentration and sizing. And Uncle is the all-in-one protein stability platform that's able to look at AAV and antibody stability very quickly and with a small sample volume in a lot of unique ways. So first, let's focus on Big Tuna, which is here to help you simplify your sample prep workflow. Because it's an automated buffer exchange platform, it means you can explore that wide world of formulation and get into those tens or hundreds or thousands of formulation protein combinations that you really want to. Beyond buffer exchange, we'll also look at how Big Tuna helps you with sample concentration with AAVs and with sample cleanup for LNPs. Big Tuna uses a filtration plate-based exchange method where we have either the unfiltered 96 or the unfiltered 24 consumable, which is like your everyday normal benchtop plate, except at the bottom it has a regenerated cellulose molecular weight cutoff filter through which Big Tuna will use positive pressure and force your liquid through to do buffer exchange and concentrate your sample inside of the wells of the unfiltered plates. And each of these consumables has a variety of molecular weight cutoff filter available, making them perfect for anything from proteins to AAV, DNA, LNPs, you name it. How Big Tuna works is through an automated pressure-based ultrafiltration dye filtration approach. So first, in each of those unfilters, Big Tuna will use an ultrasonic contactless system to measure the volume in each well. From there, it will use positive pressure with mixing to force solvent through that molecular wave cutoff filter, and then it will measure volume again to know how much volume has been removed from your sample. From there, it's just a matter of adding in your new buffer with a refill and repeat until you achieve the desired final volume and concentration and percent buffer exchange that you really want. So we like to say that Big Tuna kind of takes care of your lab chores so you can go and do the more important stuff in lab. So in terms of proteins and antibodies, those sample prep challenges that we talked about, we're exploring the formulation space. Let's take a look at how Big Tuna helps you do that. Well, because formulation is a lot to take on, and the more formulations you add, the more hands-on you have to be in lab, having an automated solution for buffer exchange really means that you can explore that whole formulation space. And in this case, we're going to take a look at crossing 4 different antibodies with 6 different excipients to understand where the best place in that formulation space may be. In this case, on the left, we're starting with 8 milliliters starting volume for each of these antibodies at about 10 milligram per ml concentration. Big Tuna will remove about 66% of that solution every round and then replace it with a new buffer until a total of 96% buffer has been exchanged. And at the end, there'll be a concentration step to concentrate each sample down to at least 50 mg per ml concentration. So at the end of this experiment, the first readout that I want to do is concentration. And that's pretty easy to check out on our proteins. Across all 4 of these different proteins and 6 different buffers, we get a nice average concentration of 53.6 mg per ml. And this is all done in a total run time of about 10 hours, 8.5 of which was the buffer exchange step, and 1.5 was that final concentration step to get from 10 to 50 mg per ml. But beyond just exchanging buffer and looking at concentration, we also have this data point from Big Tuna where we get a little bit of extra insight into developability. As these buffers have been exchanged, and here we're concentrating just on MAB4, as it gets more and more exchanged into the intended buffer, we can track the flow rate in that buffer. And what we're seeing is for 3 of the buffers, the starting buffer, and when we added mannitol and glycine, we get high flow rates. Sucrose gives us an intermediate flow rate, and arginine gives us the worst flow rate. So these actually have a bit of information to tell us about the manufacturability, viscosity, and injectability of each of these molecules. So something we can pay attention to and actually use as an output from the Big Tuna experiment. But past that, we want to take a look at stability, because that's also, the critical measurement for when we put an antibody into a new formulation. So for that, we'll be using UNCLE, which is our all-in-one protein stability platform. It has full spectrum fluorescence, which those reads will give you the whole picture of your protein behavior, whether you're using protein intrinsic fluorescence or dye-based fluorescence. Static light scattering, or SLS, will be able to detect when particles are formed, and with 2 different SLS laser wavelengths, you can look for small or large particles at the same time. And DLS, or dynamic light scattering, is going to help you crank up the detail on sizing and really understand the size at the start and end of your experiment and see what has really happened with aggregation. Apply these 3 different detection methods across a thermal ramp, and you'll be able to understand stability of your antibodies in these different excipients. So how a readout on UNCLE can look is here, again, focusing on MAB4, where we're tracking the BCM wavelength, which is, put simply, a measure of the red shift of protein unfolding. So as proteins unfold, that BCM wavelength will increase. And we really care about inflection points in that curve. In this case, what we're seeing is, well, first, arginine, the arginine formulation, has the signal increase first and has the first inflection point with a melting temperature at that inflection point of 68.8 degrees Celsius. But really, that means that we've made our antibody least stable in arginine. What we want to focus on is the buffers where the protein's most stable. And we see that in 3 different excipients, the red, light green, and yellow ones, which are sucrose, mannitol, and glycine, where in each case we've increased the melting temperature, and you'll see those BCM increases all the way on the right of this curve. So those are the most stable proteins, or the most stable formulations for this protein. Now if we compare our 2 pieces of data here, the flow rate information and the melting temperature information, we're able to select excipients that both improve stability and improve flow rate. So if we're only focusing on melting temperature, glycine, mannitol, and sucrose would be the winners, but we know from our Big Tuna readout that sucrose actually impaired flow rate quite a bit, so glycine and mannitol might be a preferred excipients out of this formulation screen. Okay, so beyond just antibodies and formulations, recently we've launched a new application for Big Tuna where you can now offload buffer exchange for gene therapies and vaccines. So Big Tuna can actually go ahead and change the setting on its laundry chore from antibodies over to AAVs. Let's take a look at that example. So there's never enough AAV. When it's manufactured, it's quite dilute, and to get to more characterization methods and ultimately to your final therapeutic dose, you need to concentrate these very dilute viral particles. So in this case, what we'll be doing is taking Big Tuna to concentrate viral particles, and it's capable of going up to 100x. And in the case study that I'll show you, it goes up to about 36x just based on how much volume we had available. So how this works, again, is by simply taking our sample and running it through a concentration program, which is able to get us to our final desired concentration and really replaces a more complex workflow where you have an ultrafiltration exchange-based method and then a concentration step. Afterwards, that's all manual. In this case, Big Tuna will automate those steps in the process and keep your hands working on more important things in lab. So how a 100x concentration workflow would go is by starting with 48 milliliters of sample that can be split across wells 1, 2, 3, and 4 here in your sample reservoir. Your goal is to have this end up at 500 microliters of volume to get to your almost 100x concentration. And the way Big Tuna will work is it will draw from those reservoirs 1, 2, 3, and 4, use that to refill sample 5, and do that buffer exchange and concentration workflow that I discussed earlier. And ultimately, you end up with a final volume where you have those 4 chambers all focus down into one chamber. And Big Tuna can do this for 24 samples at once, so you can run 24 different samples in parallel if you want, or 24 different instances of the same sample. So our case study for this is going to be concentrating dilute AAV by 36-fold, and we'll use Stunner as the analytical instrument to check the experiment, which is going to give us a good readout on AAV capsid titer and empty full. So explaining a little bit more about Stunner is its UV-Vis and DLS methods give you the complete AAV story. So beyond capsid titer, you also get empty full ratio and aggregation information using only a 2 microliter sample in a read that takes less than 1 minute per sample and without any kind of labels, reagents, or standards on your plate. You simply just add your AAV and the UV and DLS take care of the rest. How this works is you drop in your 2 microliter sample into our microfluidic circuit. That's kind of like a well in a 96 well plate. That sample is wicked into the chamber, into that serpentine microfluidic reservoir, where it hangs up until you put the plate into the stunner, and the sample is sucked into those circled 2 diamond-shaped microfluidic cuvettes there, or microcuvettes, where they have 2 different fixed path lengths that allow us to do incredibly precise UV-Vis reads. They give us very, very precise readings on concentration. We'll also do a DLS read in one of those chambers. So on the left, we get an example of DLS and SLS information from AAV. So this will tell us that our intact capsids are around 30 nanometers in diameter, while our aggregates are the gray peak at around 150, 300 nanometers in diameter. And we'll use this information to focus just on the static light scattering intensity information. That'll help us get a good read on concentration. For UV-Vis, we're getting a total spectrum absorbance read. That's the gray line on the graph. And then we're able to deconvolute that into the signal coming from just the DNA component of AAV and the signals coming from just the protein component of AAV. Combine all those pieces of information together, and we get a complete titer story. So starting from the protein world on the left here in blue, the total height of that bar actually comes from the UV-Vis, so we know how much protein is in our sample. So that's how much protein is shown here in this blue bar on the left. But because we can combine that UV-Vis protein information with the light scattering information coming from DLS and SLS, basically telling us how brightly the capsids are shining when you bounce a laser off of them, tells us a lot of information about concentration. So if we combine that light scatter information from DLS and SLS with the total protein amount from UV-Vis, and we can understand how many capsids we have and how many protein aggregates we have. So that's how we're able to split up that total protein amount into smaller, buckets for aggregates and capsids, is based on how much light scatter information we're getting from our sample. The green bar on the right is the DNA world, and again we're starting with the total amount of DNA from the UV-Vis read, and then our light scattering information gives us more information about what's coming from a full capsid, how much could be there scattering light, and we'll use that information to split up that green bar into extra DNA floating around and DNA that's packaged inside of capsids. So all that together helps us get really, really accurate information on capsid titer. So in this case, we're comparing AAV capsid titer for full and empty AAV9s on the y-axis for Stunner against the x-axis for a AAV9 capsid ELISA. So something that's going to take a very hands-on approach and a couple hours to do. But in Stunner's case, it's simply pipetting on your samples, take no further action in terms of standards or reagents, and then get a read on those 64 measurements in less than 1 hour on Stunner. So using that Stunner analysis method with our Big Tuna concentration experiment I described earlier, we were able to get a read on the initial AAV concentration in capsids per mil. We know we concentrated that to a target 36x concentration would have been about 1.7 E13 capsids per mil, and we've exceeded that a little bit to get to 1.9 E13 capsids per mil. We've done this by decreasing our sample volume from 18 milliliters per sample to 500 microliters per sample, or to 524 microliters per sample. And as a quick check of sample quality afterwards, we know that we've not impaired our empty full ratio for our AAV. In fact, we've refined it a little bit so that we have slightly more full capsids as a proportion of the total amount of sample in the AAV at the end of the experiment. And actually, this whole concentration experiment only took about an hour and a half to do, cycling from that 18 milliliters of volume all the way down to that 500 microliters of volume. Okay, so now let's go to the last Big Tuna example and turn the dial from AAV over to LNPs. In this case, LNP sample prep challenge was to remove organic solvent after production. So how LNPs are typically made is by starting with an aqueous phase containing your payload, and an organic phase. You're going to combine them and make an emulsion that starts to contain your LNPs. But then you do need to eliminate that organic phase afterwards, and Big Tuna is going to take care of that through some buffer exchange. So here we're using an mRNA LNP, and we're going to have it starting in 10% ethanol and PBS. We'll remove that ethanol and concentrate it threefold. We're doing this in a 100 kilodalton unfiltered 24 well plate. So, and again, we'll do the analysis on Stunner. So very quickly explaining Stunner's approach to LNP analysis. Because we have our very, very short path length microcuvettes, we're able to get UV-vis reads on these very turbid LNP samples. So where classical UV-vis reads might struggle with the very turbid LNP samples, Stunner's short path length mean that it can still see through those samples and get accurate UV-vis reads. So what's happening there is we'll have raw data of UV-vis absorbance on the left, and Stunner is able to deconvolute that to what we see on the right. So at the top dark gray line is our total spectrum. Stunner is able to match that to the turbidity signal, shown in light gray, the signal just from RNA, shown in green, and the signal from lipids and buffer components, and actually extract out from that sort of very noisy raw data signal the actual concentration of RNA present in our sample. So we'll use this to take a look at what the concentration of RNA is during our Big Tuna experiment. In this case, after 90 minutes of buffer exchange and concentration, we're starting from an initial RNA concentration of 26 microgram per mil, and our target at a 3x concentration was 78. We've met that and gotten to 83.5 microgram per mil concentration. We've maintained encapsulation efficiency, hit our target volumes, and hit our target percent exchange for how much buffer and how much ethanol we're getting rid of in our solvent. So pretty nice and easy example of buffer exchange on LNPs in terms of concentrating them and removing that ethanol. And that's how Big Tuna, Stunner, and Uncle can help you solve your sample prep challenges on sort of the latest cutting edge biomolecules, whether those be from monoclonal antibodies, AAVs, and other viral vectors, and LNPs. And now I'll ask if you have any questions. And if you think of something later, please feel free to contact us at info@unchainedlabs.com.